just in case you're wondering what the method is behind all of this madness. So within OpenStax, you're going to want to access Chapter 3. There may be an item or two that relates back to what we did in Chapter 2. So you may find yourself going back into Chapter 2 for a few things here and there. I doubt it, but you might. For the most part, what you're going to need is in this slide presentation, as well as within Chapter 3 of OpenStax. So when we look at the anatomy of cells, we're going to look at each of these cell parts as not only how it's structured, but also a little bit in regards to what it does. So we call that the functional anatomy of cells. So we generally, again, won't get into too much physiological biochemistry, just a little bit. So a typical cell, what we see and what we're going to see on the next slide, is called a composite cell. This next slide of the composite cell is simply kind of like the golden corral of cells. It's all of the possibilities. So you've got the chocolate fountain, you've got the pizza station, the sun ice cream. It's around lunchtime, guys. But so anyway, functional anatomy of cells, that typical cell we see in an illustrated kind of is just everything possible. Okay, no cells are going to look like this. So that's why they call it a composite. All cells are going to vary in structure and function. So, and again, that's going to be dependent on what organ and what tissue we have, what kind of combination. So in other words, a heart is going to have specific cells to the heart, whereas the brain is going to have a different setup regarding cell structures and cell functions than the heart. So brain and heart, different organs, different cell structures, different functions. So red blood cells, we're going to see, don't have a nucleus. Most of the cells in our body are going to have a nucleus. When we get to that spot, I'll bring that up again. But again, you want to, as we look at this next slide and we look at all of these cell structures, again, not all cells have these structures. Several of them do. Furthermore, not all cells have the same maybe amounts of these structures or the shapes of these structures. So all of these cells are going to have some sort of cytoplasm or fluid kind of matrix inside of the cell. You know, that's going to differ from cell to cell. So anyhow, just keep that in mind. Cells are very different. Cells are specialized depending on what the function of the organ ultimately is. And of course, we're still dealing with the micro level, the microscopic level, just like we saw with chemistry. Next week, we're going to stay in that micro level for just the beginning of our discussion, and then we're going to get into our first organ system, which is going to be skin. So we're going to turn things into the macro scale, or the gross anatomy aspects, beginning next week. So if cells are boring for you, I do apologize. I'll try to inject some spark to it, but and if chemistry was not really your thing either, well, just hang in there. You've only got another week or so, and then we'll jump into the big scale stuff: skin, and then bones, and then muscles, and then glands, and hormones, and nervous system, and blood, and heart, and guts, and all that good stuff. So you guys have made it through the first 2 weeks, now 3 weeks of kind of that tiny nitty gritty stuff. So again, good work, and keep it up because we will see this throughout. Whenever we discuss new organ systems, we will look at some of the specialized cell structural mechanisms that we may find. So if some of this is a little new and a little strange in the wording. Again, don't worry, because you'll see all of this again. We're building that giant puzzle that I keep reminding you of. So here's our composite cell. We see a lot of pretty colors and a lot of things going on there. And I think all of you at some point in your lives have had experience with cells. And you do recall that we are going to have a cell membrane typically. We're also, which is kind of like the skin of the cell. We're going to have a nucleus of a cell, which kind of plays the role of the brain, where we find the DNA and the genetic code, kind of the blueprint and the jobs and the all of the roles that all of these other cellular structures and organelles are supposed to be doing. All of those directives and those kind of 3-ring binders, if you will, not nowadays. Now the jump drives, flash drives, these storage containers are all located in that nucleus that are going to be basically a series of recipes with different chemicals on these recipes and different amounts and ratios, and when the cell organelles need to produce a specific enzyme or chemical or a specific structure, or they need to create some sort of antibody. A lot of that information is going to be stored in these cookbooks that are within the nucleus of our cells. So generally speaking, we're going to see a nucleus in all of our cells in the body. I did mention the red blood cell doesn't generally, we don't see a nucleus there. But so yeah, nucleus and cell membrane, I think all of you have heard of those at some point. We do have that fluid matrix that everything's kind of floating and suspended in. We call that cytoplasm. So we'll look at that a little bit later. It's kind of all that negative space. We see all of this pretty purple folds and curves and light blues and greens and yellows and little circles, yellow dots. So in between all of those positive space structures is what we call negative space. And that negative space, you can see more of it up here. It's kind of an orangey, yellowish brown color. That's called cytoplasm. So we'll look at that. It's kind of like the blood of the cell. So anyway, we see mitochondria, little bean-shaped items scattered throughout. So we'll take a peek at those in more detail. We see little spikes and spindles. So there's almost kind of like a skeleton, different skeletal structures and within the cell, different tubes and filaments that we'll take a look at. And then we've got all these little folds and tubes. These are kind of like the factories of the cell. A lot of our synthesis and chemical reactions that are occurring inside of a cell are going to be found in these kind of rooms and these folds. And then lastly, we've got kind of a digestive mechanism that we'll look at that helps to eliminate some of the waste. It helps to pick up and transport some of the products that are being produced in these factories. So we'll take a peek at the Golgi apparatus along the way too. So now we see some little tails and little kind of hairs and spikes. So we'll also look at those. Okay. Generally speaking, the big items that we look at, I just mentioned the membrane, the plasma membrane, and then the cytoplasm of the cell. And then we've got all those little organelles, all those little tubes and circles and globes that are cruising around and floating in that cytoplasm. And then we've got, of course, the nucleus and then the skeleton. So we're going to take a peek at each of these in a little more detail in these next couple of slides. When we look at the cell membrane, we generally are thinking of that outer membrane. kind of like not really the cell wall. We think of maybe plants as having more of a cell wall, to be maybe a little bit more impenetrable, and provide a little bit more rigidity, um, to contain fluids. The cell membrane goes beyond just this outer kind of skin layer. So the first slide gets into cell membranes and discusses different organelles and different structures also have membranes. So we will look at not only the plasma membrane, but some of these organelles are also going to have membranes. So we'll keep that in mind kind of as we go. That we have what are called membranous organelles. So the first, we have this fluid mosaic model. I don't think those are two terms that you'll ever probably hear again, but this is just what they call this kind of representation or model of the cell, that we have a mosaic, meaning we've got a lot of different patterns of structures and fluids in which these structures are suspended. So that's where they get that fluid mosaic. It's ever changing. There's a lot of flexibility involved. It's not a rigid structure. That's also where they come up with this terminology. When we look at the outer membrane of the cell, the actual cell membrane, we see it represented as more than just, again, a wall of structures that don't allow anything in or out of the cell. The idea regarding cells is we need to feed them. Really what we consume is going to ultimately go out to our cells. So the delivery of water, the delivery of nutrients has to occur via a semi-permeable membrane. So we do see, for the most part, our cells throughout the body are going to have a membrane that is going to be somewhat permeable. Typically, the structural feature of a cell membrane is going to be lipid based. So it's going to have what we call phosphate. So the P on the periodic table, we also see phosphate or phosphorus coming up when we look at the ATP energy molecules. So P comes up frequently throughout both general and advanced AMP. But ATP, the P is for phosphate. We see phosphate again when we look at the cell membrane. Okay, we call it a phospholipid bilayer. And I think maybe some of you have heard that term, and I think maybe all of you came across it either in week 1 or week 2. But there it is again, phospholipid, lipid meaning fat based. So if you're ever asked about, the cell membrane, what macromolecule is the primary macromolecule of the cell membrane, you're thinking lipids or fats. Okay. Now, embedded within that lipid membrane are going to be proteins. So we see little, we'll call them membrane proteins here in a little while, and look, or we just call them that, I guess, but we'll look at them and see it in more detail here in a second. But we do see some channels to allow certain items like sodium and potassium, chloride ions to pass through the cells. We also will see what we call pumps. So some of these proteins are going to be a type of pump that we're going to have to move some sort of ion against its concentration gradient, which again we'll look at with cell transport here coming up. You can also see we've got some examples of glycoprotein and glycolipids. a couple of terms that you saw in your portfolio last week. These glycoproteins and glycolipids. Typically, a glycolipid is going to be kind of a chain of sugars. You can see these little, they're kind of, we see some pentose or five-sided sugars. We see some six-sided sugars all connected. This is going to tell us this is some sort of sugar, chain of glucose or pentose sugars. And notice it's directly attached to the cell membrane, which again is lipid-based. So we would call this maybe a glycolipid. And then over here, we've got an integral membrane protein, and it's got a little chain of sugars attached to it. So we could call this little chain glycoprotein, or this little unit, a glycoprotein. We can see it again over here. Okay. And this, by the way, is the outside of the cell. We would call this area the extracellular space, and it's typically comprised of fluid. Generally, we would see water in that area. Okay, so outside of our cells, and then inside of the cell down here is what we call cytoplasm. And that cytoplasm is also a watery environment. And in fact, a high percentage of the water inside of your body is going to be intracellular or inside of your cells. Okay, so anyway, there's the cell membrane. It's a phospholipid bilayer, meaning we've got two layers. You can see we've got kind of a double layer of molecules going on here. We call this a bilayer, phosphates and fats or lipids. So this area here in the middle, this would be what we consider to be hydrophobic. Water generally doesn't get along well with fat, so we would see this being a hydrophobic region. These so the tails are hydrophobic, and then these heads, you can see the little balls here, these are the heads. They're going to be what we call hydrophilic, so they like water. They, in fact, are living and immersed in water, both the inner aspects and the outer aspects. So we do have, again, these hydrophilic or water-loving heads of this phospholipid bilayer. Okay, and then the hydrophobic. So in order to we do get water going in and out of a cell. It's going on all of the time. It has to happen. So we do have what we call pores or channels for water to be able to pass through. Okay, and you can see little cholesterol molecules. We, of course, relate that to lipids or fats as well. So you can see these little cholesterol chains in there, and they're going to provide some structural support, certainly. By the way, too, these carbs or these glycos that are attached, sugar and glucose, we typically want to always relate that to energy. So these are on hand possibly because this is maybe like a pump of some sort. So we're going to need ATP or energy availability. Therefore, we need glucose availability. So that's usually when you see carbohydrate or gluco or glyco somewhere, you're thinking that there's a need for energy. And we're going to look here in a few minutes at the main organelle inside of the cells that's responsible for working with that glucose and getting that energy going, or that ATP going. Okay, so here are another way of looking at those membrane proteins. They call those IMPs. I can't make this stuff up, right? Integral membrane proteins. Okay, and again, some have carbs attached to them. Some are going to be receptor sites as well. So some of these are going to be related to antigens and antigen binding sites, enzymatic binding sites, hormone or neurotransmitter binding sites. Typically, if it's a structure in the body, it's going to be protein based. So the cell membrane, of course, is lipid, but it has the protein structures embedded in it. When we look inside of a cell, we're going to see a whole bunch of different structures. These structures are going to be protein based. Okay, so keep that in mind as well. Structures typically are going to be protein based, microstructures. So let's move on from the cell membrane. We'll see it again shortly when we get into cell transport mechanisms. All right, cytoplasm is next. Again, that's that fluid within the cell. It's literally broken down, means cell plasma or cell fluid. And typically plasma is a fluid that isn't water exclusively. It's a fluid that's kind of like a solution or a suspension. It's got some sort of structural aspect, suspended or floating, swimming, immersed, however you want to put it, within that fluid. Cytosol is kind of the water of the cytoplasm. So cytosol is not necessarily considered to be cytoplasm. Cytosol is the fluid aspect of the cytoplasm. The other aspects are going to be organelles, these little microstructures, again, cytoskeleton, the nucleus, all of these structures that are within the cell are going to be kind of floating, suspended, swimming, immersed in that cytosol. The whole group is called cytoplasm. So here's just an illustration of what we would maybe see with cytoplasm. We see messenger RNA, we see ribosomal RNA. At the moment, that doesn't really mean anything to you, but RNA, we'll leave it at that for now. We do see other proteins, other protein bodies, proteasomes in here as well. Little microtubules. So all of the negative space, the areas that don't have color, that you don't see, that's going to be what we call the cytosol. Okay. So they don't label the cytosol. All right. So let's move on from cytoplasm. So cell membrane, cytoplasm. Now we're going to get into the little more of the specifics with organelles. Remember when we mentioned the cell membrane, of course, is going to be membranous. We also are going to see several of the organelles inside of the cell that also have membranes. In fact, the majority of these larger organelles that we look at are going to be membranous. They're going to have sacs or canals. There's going to be some sort of a lumen of the tube, making it membranous. And I label them throughout. I've got a little chart so you can see which ones are which. Okay, non-membranous are going to be more protein-based, more solid. This is almost like the hollow organs of the body versus the solid organs of the body. So cells are just kind of like little bodies, basically. So the membranous structures or organelles are going to be, again, the ones that we're probably going to spend the majority of time on. The nucleus, the ER or endoplasmic reticulum, the Golgi. We'll look at vesicles and vacuoles and lysosomes that come off of peroxisomes that come off of the Golgi, as well as mitochondria. We don't deal with chloroplasts for human. Okay. Ribosomes, cytoskeleton, centrioles, and cilia and flagella. These are going to be non-membranous. They're more protein, solid based structures. They're going to provide, again, more structure for the cell versus tubes for transporting items in the cell. So these are maybe more related to like the digestive system and the cardiovascular systems of our body. These are more related to the muscle system and the bone musculoskeletal system. Okay, so let's look at a couple of these organelles. We see the first couple of membranous organelles we call endoplasmic reticulum. We abbreviate that as ER. And when we look at the endoplasmic reticulum, we're going to see two different types. The first type is called rough ER. Okay, rough ER. What makes the ER endoplasmic? So let's break that word down really quickly. Endo means inside. Plasmic has to do with the cytoplasm. So it's kind of redundant, I suppose, because we know we're dealing with items inside the cytoplasm. We're dealing with items inside the cell. So don't let the big words get muddying your brain. Endoplasmic just means inside the cytoplasm, inside the plasm. We already know that. We know we're dealing with something inside the cytoplasm. Reticulum has to do with folded. When we see reticular, we're dealing with some sort of a folded appearance. So remember, we have membranous organelle here. So we are going to have kind of a tubular appearance. There's going to be a lumen or kind of the inside of the tube, and it's going to be folded. The tubes are folded, so they call them reticular. So inside the cytoplasm, we have folded membranes. That's what this means, and so we that's a lot. It's a mouthful, so we call it endoplasmic reticulum. That's a mouthful, so we break it down even further and just call it ER. And that ER can be rough or smooth. Rough means it has ribosomes, and we haven't looked at ribosomes yet, but when we do in more detail, we're going to think back to the rough ER as well. Ribosomes always relate to protein. Now, as we analyze some of these cell structures, really we're just going to look at carbohydrates, fats or lipids, and proteins. What are the structures of our cells, or the organelles of the cells, going to use? Well, to either synthesize or catalyze, to do something with. Because remember, we're giving the cells all of these macromolecules and breaking them down and allowing our cells to then do something with them. Well, the protein part, so this is why you eat protein, is so ribosomes can work with and synthesize some of these amino acids to become enzymes, or to become, remember we learned enzymes last week, are protein based. So proteins and amino acids can be strung together in a certain order to produce specific enzymes. And the recipe books, the cookbooks for all of those enzymes, come from the nucleus. So ribosomes and how they work with, they basically work with proteins by stringing together amino acids. And remember, amino acids are the small building blocks of proteins. They're basically kind of like a set of beads. If you remember and some of you may still have them. I do. They're not mine though. They're my daughter's. They're kind of like little tackle boxes full of different beads, different shapes of beads, different colors of beads. Each of those beads could represent a different amino acid. And so if I take some sort of string or twine, or they've got that elastic kind of flexible stuff that comes inside of those kits, so you can start stringing these beads in a different order. Or you can just, like they just string a bunch of pinks together, or there may be a pink and then a purple and a pink and a purple. So regardless, no matter what they produce, they're probably going to have a different arrangement, a different order, and each bracelet, each necklace, whatever it might be, is going to look slightly different. So it may have a different function. So that's basically what ribosomes are doing, is helping to string together they kind of help to read the code that they read the recipe that the nucleus has sent out into the cytoplasm. The ribosomes kind of help read that recipe and kind of enable a few of the sous chefs to help put these or the apprentices to put these beads in the right order to get the right type of necklace, or the right type of bracelet, or the right type of enzyme, the right type of widget, however you want to relate it. But the rough ER is basically the factory where proteins are getting put together. Protein synthesis, where all of these amino acids, all these beads, all of these parts are being assembled based on the directives given from the nucleus to the ribosomes, and again, they need a place to put together some of these structures or some of these enzymes, and the rough ER is the factory where that can happen. So anyway, that's what's going on there. The rough ER is typically going to be very tightly connected with the nucleus. I'm going to go backwards here. We typically see the nucleus here. We'll see that rough ER, all of these kind of this network of folded tubes is going to be right up against the nucleus. We can see all of these little yellowish circles on top of that ER. Those are the ribosomes, and those ribosomes attached to the endoplasmic reticulum are what make this a rough endoplasmic reticulum. Okay, so this is a rough endoplasmic reticulum because of the presence of ribosomes, which both then the ribosomes are going to enable protein synthesis to occur, and then the endoplasmic reticulum is going to be kind of like the location. It's like the factory where this can happen. And then we see we go out a little further, and we see another kind of group of folded membranes that appear to be kind of stacked and organized a little bit. They don't look much different, except we're lacking all of those ribosomes. So we call this batch of endoplasmic reticulum smooth ER. Okay, so rough ER has ribosomes and works with protein synthesis. Smooth ER lacks ribosomes and deals with lipid or fat synthesis. And you'll recall the cell membrane is primarily lipid-based. So we've got a connection with the cell membrane where lipids are and that smooth ER. Okay. Rough ER, smooth ER, protein synthesis, lipid synthesis. Okay, and ribosomes being present make this rough ER. No ribosomes present, smooth ER. Lipid synthesis closer to where we need lipids, right? That phospholipid bilayer. And then lastly, the ribosomes were protein synthesis. And again, those directives, those recipes are coming right out. They're photocopying them, transcribing them inside the nucleus, and we're delivering those recipes, and then we're translating those recipes out here, either in the cytoplasm with the help of the ribosomes, or if we've got maybe a little bigger job or a little different type of job, we can do some of that assembly inside that rough ER. All right. So anyway, okay. So ER is membranous, rough ER proteins, smooth ER lipid. Those ribosomes we just mentioned that make ER rough are non-membranous. These are tiny little buggers that are scattered all over the within the cytoplasm. So they're not only attached to the endoplasmic reticulum, making it rough, they're also swimming around and floating around and cruising around freely within the cytoplasm. And again, they're responsible for assembling proteins or assembling amino acids to get proteins. Okay, so a protein is simply a bunch of amino acids strung together, and that's what the ribosomes help with. Okay, and again, the the endoplasmic reticulum ribosomes, that's like a factory. They're producing things that are more than likely going to be exported out of the cell. Now, not always. They could, some of the items that the rough ER is producing, or not really producing, synthesizing, or those beads are getting strung together. Some of that stuff's going to stay inside of the cell. We have basically three options. What's going to happen to the necklace? What's going to happen to the bracelet? What's going to happen to the enzyme? What happens to the constructed, assembled protein? Well, we can either ship it out of the cell, and for the most part, the ER is making things for export. The second thing we could do is not necessarily export them, but send them to the cell membrane. So these proteins, or these bracelets or whatever that were produced or assembled, I should say, maybe their destination is as an integral membrane protein. So they're not going to get shipped out. They're going to get sent and embedded into the cell membrane. So they become an IMP, an integral membrane protein. Hey, and then the last destination possibility for a protein that's been synthesized is to stay in the cytoplasm. Okay, so we could just keep it in the cytoplasm, keep it inside of the cell. So it could go extracellular, stay in the cell, or be intracellular, or it could become embedded in the cell membrane. So anything that's assembled in the cells by these ribosomes can end up in one of three places. Okay. If it's a free ribosome that's just kind of swimming and floating around, they're going to be putting together bracelets and necklaces with these amino acids that are probably going to stay inside of the cell. They're probably going to be for the cell's use. These could be an enzyme, some sort of intracellular and inside of the cell enzyme. Okay, so anyhow, ribosomes, protein synthesis. If we're going to make something a little bit more bulky and hefty, maybe it needs to get shipped out, like a collagen fiber or something. Maybe we need an enzyme that needs to get shipped out. Maybe we need an integral membrane protein. We need a new channel. We need a new pump. Our pump is worn out. So the rough ER is going to be the site of assembling the integral membrane protein. Or maybe we simply just need some sort of enzyme for use inside of the cell, or we need some sort of microfilament for inside of the cell to repair a cytoskeletal issue. All of that is possible. We're either going to keep the assembled amino acids in the cell, ship them out of the cell, or embed them in the membrane. So anyhow, here's what ribosomes have: small and large subunits, and again, basically the messenger RNA. So RNA is a sort of like a single-stranded photocopy of DNA from the nucleus. It's, in essence, it's a photocopied recipe from whatever the nucleus needs to have created or made. Okay, so we kind of unzip a piece of DNA, make a copy. We call that transcribing or transcription. We're going to send the messenger. The mRNA is basically the messenger that has the photocopied recipe. And that messenger RNA leaves the nucleus with the recipe or the blueprint or whatever we want to call it, the instruction key or the assemble guide, and it's going to run it out to the ribosomes, and then the ribosomes are going to start kind of replicating and photocopying and basically translating what this is. So ribosomes are kind of like they basically are the factory kind of super or the boss of the factory. So they're going to get directives from the owner of the company saying we need 1,000 of this widget produced, 1,000 of these bracelets produced, 1,000 of these enzymes produced. So here's the work order basically, and that's what the messenger RNA is carrying: the work order, the blueprint, the recipe of what the nucleus needs for the better of the cell and the health of the cell. Okay. And it's the ribosomes that basically translate that. They read it and say, all right, everybody, this is what the boss wants. Let's put together. And so here we have the exit hole. And now we've got what we call a polypeptide or a long chain of amino acids. Okay, so we've got the code. We put all of these little codes together, and then out comes the widget. Okay. So that's what ribosomes are doing. And again, this polypeptide is a protein. Okay, it's a protein. We see transfer RNA. It's just basically the messenger brings in the blueprint. The transfer RNA is going to help with the assembly of and reading of that the directives of the work order and get everything organized and transferred over into what the nucleus wants. Okay, and then we've got the Golgi apparatus that's also a membranous organelle. Typically, the Golgi is going to be, the folds are going to be a little bit curved. They're going to be almost parallel with one another. There's somewhat of a, let me go back to our. Okay, so we see kind of of an arrangement with the Golgi, even under an electron microscope. When we look at the Golgi, the folds and these layers are very close together. It's usually kind of huddled more out towards the cell membrane. It's usually a little further away from the nucleus and the rough ER. And what the Golgi does is it basically is the packaging center. So if the rough ER is the assembling factory, the Golgi is kind of like the UPS hub or the Amazon warehouse, or it's kind of like the warehouse. It's the shipping and packaging center. So we make the widgets or the necklaces over here, and then we drive them using one of these little vehicles. They're called vesicles, but if you take the S out of there and you put an H in, you have the word vehicle. So a vesicle is basically a vehicle that's pinched off. It's kind of like it's basically a UPS truck, or nowadays an Amazon semi that's cruising around, going from Golgi apparatus over to the rough ER, or it can go to the smooth ER. Okay, but it's basically picking up the finished product and then delivering it wherever it needs to go. So these are called transport vesicles. They're little buds pinched off of the Golgi apparatus. This is the ship packaging and shipping department. And the vesicles are the vehicles that are zooming around back and forth carrying the shipment. The Golgi also has a few other jobs. It does a lot of packaging of waste. So we have other items, what are called lysosomes and peroxisomes. Lysosomes go around and kind of clean up the garbage, and they're kind of the janitorial or building maintenance guys that and gals that go around and kind of clean things up. So the lysosomes keep things relatively tidy inside of a cell, and they're going to be parts of and aspects of the Golgi as well. And then peroxisomes, think of like peroxide, like you may have in your medicine cabinet. Rubbing alcohol and hydrogen peroxide are two items that can be used to kind of clean up wounds, right? Kind of an antiseptic property. And these peroxisomes are also aspects of the cleanliness of a cell, but they're more they release more specific enzymes for fighting any type of pathogens that may be entered. So if we've got some bacteria or viral material inside of the cell, peroxisomes oftentimes are able then to release some sort of specialized chemical, an enzyme typically, that's going to speed up the rate of reaction to basically disinfect the area. So lysosomes, peroxisomes, vesicles all work with the Golgi apparatus. They also work with the cell membrane. So when the cells bring things in, or when the cells excrete items, oftentimes we see the fusion of these vesicles. So sometimes the vesicles are not only going back and forth between Golgi and the factory, picking up the product, the finished product, and for shipment and packaging and shipment, but they also, these vesicles, some of them are garbage trucks. Like I say, these lysosomes and vesicles can be more specialized to transport waste out to the cell membrane for what we call exit or exocytosis. Okay. So anyway, that's what the Golgi is doing. Let me get back to our, so again, it processes items from the ER. We have what are called cisternae, and that simply means these are these little folds or cisterns. And then this is the lumen. Remember, this is a membranous organelle. So we do see a lumen of that tube. And then again, the cisternae or the cisterns. Those are what form, those are the structural aspects of the Golgi that form the folds, and then the lumen is the inner portion. Then we can see these vesicles, these little vehicles that kind of pinch off and cruise around. So you, again, usually under a microscope, a Golgi is going to be a bunch of compressed, very tightly together, whereas smooth ER and rough ER aren't as condensed like this. You can see there's a lot more spacing typically. Okay, so we have secretory vessels. These are going to be for secreting enzymes or secreting chemicals or secreting waste for elimination out of the cell. And again, I mentioned lysosomes. These are going to be membranous. They're little, kind of little trucks that come off of the Golgi apparatus. Okay, they're little sacs that pinch off. They kind of digest waste. They're like garbage compacting vehicles. Hey, lysosomes. So they help to keep things clean. You can see we've got some cellular debris. These guys just come in and envelop that debris and seal everything up. And then we get kind of this residual body. This is going to be kind of like the garbage or the waste or the poop that comes off and gets, usually it's going to go through what we call exocytosis or elimination from the cell. Okay, so lysosomes help take care of a lot of garbage and waste pickup. Peroxisomes, again, they're going to be more specific to detoxification. So we see a lot of peroxisomes. So the cells of the liver and the cells of the kidneys are going to be more loaded with peroxisomes. when we see that an organelle participates in detoxification, we if you know that the liver and the kidneys are the primary organs of the body that detoxify, a lot of you probably already know that, right? You either heard it, you work in the medical establishment, and you know that the kidneys and the liver are part of detoxifying. They're the primary filters, right, of the body. The lungs help too with some of that weight. So we would see then more peroxisomes located in the cells of those organs. All right, mitochondria are next. We've got the powerhouses or the power plants of the cell. And again, these guys are going to be responsible for glucose or the sugars aspect, right? We did proteins already with ribosomes and rough ER. We did lipids with smooth ER. And now we're doing mitochondria and sugar or glucose. So cellular respiration is the ability of your cells to be able to generate some sort of energy. So mitochondria are able to generate energy, and they do that through several chemical processes that we look at in more detail in advanced, but it does involve adding oxygen to the mix. So when I add oxygen, I can get, well, we call it respiration for a reason, right? Oxygen's going to be involved. Respiration just means exchanging gases. So oxygen and carbon dioxide are getting exchanged. Okay? So oxygen coming in, cellular respiration. And this is going on with the mitochondria. We're going to look at the very end, we're going to look at anaerobic versus aerobic. Aerobic simply means we're using oxygen. So the aerobic respiration is what we see here. This is going to generate the most molecules of ATP or energy. Okay. We can also do what's called anaerobic respiration. That does not involve oxygen. So AN in front of a word oftentimes means without. Okay. So without oxygen, anaerobic. Okay. That's going to take place outside of the mitochondria. Anaerobic respiration is what we call glycolysis, which takes place in the cytoplasm of the cell. It's kind of the precursor to cellular respiration. So, and we know this happens when we're at what we call an oxygen debt. We're working out, we're not used to working out, you know, go out and I start raking leaves this fall, and I haven't really done that action of raking leaves. And so my muscles are in an oxygen debt. And so they still work though, right? But I might be a little sore later on that day or in the morning. And that's due to lactic acid buildup, which comes from anaerobic respiration or using generating ATP, but not using oxygen to do it. So you do get a penalty for not using oxygen, and that's lactic acid typically. Plus, you just don't get nearly as much ATP. It's basically, it's like almost like a 2 to 36 difference. You know, you get a couple of ATP from anaerobic respiration. You can get up to like 36 ATP when we add oxygen to the mix. So anyway, my point is when you see mitochondria, you always want to think of these three letters, ATP, which relate to glucose and oxygen being converted into energy. Okay. This long stuff here, oxidative phosphorylation, just with a P, phosphate, phosphorylation, adding phosphates together. Oxidative means we're putting oxygen. So when I add oxygen, I get, I generate ATP. Okay. All right, and we get that again by oxidizing the food we eat. So we get oxygen into the cells not only by drinking water, we get oxygen to cells, but by breathing, as well as certainly from the foods we consume. We get the ingredients needed to perform that action. So anyway, we look at a mitochondria. We see that it's membranous, kind of a double-layered membrane. We see what are called cristae, which are internal folds. Then we have what's called the matrix on the inside. By the way, too, mitochondria can they possess their own DNA. So mitochondrial DNA is slightly different than what you'd see inside of a nucleus and what we call nuclear DNA. So they're almost like cells inside of our cells. Okay, so mitochondria have DNA, which is again a genetic code. Speaking of genetic codes, then that's our segue to the nucleus. So I want to look at the nucleus for just a few minutes. We do have kind of a double layer of action going on. The nucleus does consist of what we call a nuclear envelope. Okay, and just like an envelope encloses a letter, the nuclear envelope encloses the nucleus. And again, a double membrane, a lot like the plasma membrane. So the nucleus is almost like a cell within our cell. Okay, so the nuclear envelope is a double layer, a double membrane. Okay, the nuclear membrane. Okay, and it's got fluid inside of it. Specifically, it's not cytoplasm, we call it nucleoplasm. And within that nucleoplasm, we're going to see the DNA, which appear as what we call chromatin, which are threads or granules in non-dividing cells. And then chromosomes, we see those are what gets split. So chromosomes are going to be in a dividing cell. So we'll see chromosomes in that early stage of cell division, okay, which we'll see shortly. Okay, so we see the structure of the nucleus, again, similar to a cell membrane. We have a nuclear membrane. We also call a nuclear envelope. It's kind of, again, a double layer. And then we also have pores, so we can get fluids and other substances in and out, and chemicals in and out of the nucleus. And then we've got all of this chromatin. That chromatin is going to be full of chromosomes. Okay, and all that chromatin is floating around and immersed and swimming in what we call nucleoplasm. So there's a fluid inside of that nucleus. And then the nucleus has its own nucleus. So this, again, this almost looks like a cell in and of itself. And then a nucleus inside of a cell. When in fact, this is a nucleus with a nucleus inside of it. So we call that the little nucleus, or the nucleolus, and that's where you're going to find the DNA stored inside of that nucleolus specifically. Okay, so we've got chromatin and nucleoplasm out here, and then in that nucleolus, that's where we're going to find all the DNA all wound together and contained until it's ready to be photocopied or transcribed, and then we start breaking that up and letting that action go on out in the nucleoplasm. There's a pore. You can see the complexity of that pore. This is inside the nucleus. This is outside the nucleus, but we're still inside of the cell, right? We're still in the cell or in the cytoplasm. Okay, so let's move on. We've got the skeleton. So that's the brain of the cell, the nucleus, and then the nucleolus. We also have the cytoskeleton. This is the skeletal framework of the cell. We have tiny protein fibers, rod-like pieces, protein strands, protein rods, protein globules. We've got several different types of proteins that help to form that cytoskeleton. It helps with locomotion or movement of the cell or of a cell. The cytoskeleton can help with simply just a structural feature. So it helps keep the cell in essence, morphed or folded or shaped into whatever shape it's supposed to be. So it helps the cell to maintain its shape. It helps the cell with movement or locomotion. Aspects of the cytoskeleton can detect changes outside of the cell. So we'll see maybe some cilia associated with that as well, or micro filaments. Okay, we're going to see three different types of cytoskeletal structures. We have the cell membrane, or we have two. We have the cell membrane out here and the cell membrane here. So this is entire aspect is the inside of the cell. So remember, all that negative space is going to be where we find the fluid or the cytoplasm. So embedded in throughout that the cytoplasm, we do find different types of tubules. So we see the largest of the cytoskeletal framework called microtubules. And then we see the smallest, which are called microfilaments. So a filament is generally considered to be smaller than a tubule. And then in between, we have what are called intermediate, in between filaments. Okay, so those are part of the cytoskeletal structure. And again, you can notice that we've got the ER, the rough ER, ribosomes, mitochondria. We've got some cellular organelles that are held up, or their positioning is maintained by the presence of these cytoskeletal structures. And again, we've got microfilaments, the smallest, and then the intermediates, and then the biggest, or the tubules. And these microfilaments, we see these when we look at muscle cells. We'll see that term microfilament or myofilament again. Okay, so filament, tiny, tiny protein fiber. Okay, we've got a supporting framework with those intermediate filaments. And then lastly, the microtubules are the thickest, and these help to move things within the cell. So they will attach to the intermediate filaments, as well as some of these other organelles. You can see the microtubules attached to the rough ER. So as they move, we can get the rough ER, for instance, to also move. So they kind of like puppet and pull some of the other organelles within the cell. And in order to do that, in order to move other items around within the cell, they have to have a little bit more strength. So they are the thickest of that group. Hey, and then we're just about done with all of these organelles. We do have what are called centrosomes. And a centrosome is going to be made up of two little centrioles. You're going to have what they call a mother or a parent centriole and a daughter centriole. They're basically, you see the word center in there. It basically has to do with bundles of tubes that form kind of a structural framework at the poles of the cell. So cells have kind of like a north pole and a south pole. There's kind of like a magnetism contained within the cells. And these centrosomes ultimately are going to release what are called spindle fibers when we look at mitosis and cell division. So we're going to look at these in a little bit more detail coming up shortly. So they basically keep the microtubules organized, and those microtubules are organized into what we call centrioles. So you can see these are the microtubules that we looked at earlier, and we bundle those together into what we call a centriole. Okay, this is a daughter centriole and a mother or parent centriole, and these two centrioles make up what we call a centrosome. And these centrosomes are going to form the pole of the cell, the north pole and the south pole. And poles, again, have to do with attractive forces and it's how we're going to ultimately enable chromosomes to connect and then line up and then ultimately divide and get two cells to form. So centrosomes are extremely important when we talk about cell division and the cell cycle and mitosis. So we'll see that coming up a little later. This I'm not too just kind of an FYI. We have other tubules. This is a microtubule, and we have little structures that are attached for locomotion. So oftentimes we see filaments and other protein structures when we're talking about movement. So this is sort of getting the cart before the horse. I view it maybe more as kind of a teaser trailer for what we're going to see when we look at some of the cellular structural detail of muscles in the musculoskeletal system, which is coming up right around week 6. Okay, so kine or kines always means, or kinese means movement. Myo means muscle. So this is just showing us we've got some muscle movement microstructures, molecular motors. And again, they're going to be protein based. And then lastly, little extensions, microvilli, which again is kind of a teaser trailer to something that we're going to see in quite a bit of detail when we get to the digestive system. These are small folds that are on the surface of larger folds. So these are microvilli, are folds on top of villi, and villi are folds as well. So we see large folds are called villi. Microvilli are the little extensions off of the cells of the villi within the intestines. So we'll see that again in more detail later. But they are little extensions. They help with enzymatic activity, brush border enzymes. They help primarily with absorption of nutrients in the intestines. And we've got the cilia and the flagella. Cilia we see a lot whenever they're like little bristle brushes that help with kind of filtration of debris and dust. And then flagella are tails that we would see maybe on the end of a sperm cell. Really about the only cells that do have a flagellum are sperm cells. Then we see cilia. This would maybe be in the respiratory tract or the lining of the respiratory tract. We see small cilia, little finger-like, hair-like projections that help with removing microparticles. And then we can see these microvilli, little finger-like projections. So microvilli are more like fingers. Cilia are more like hair, and flagella are like tails. So that's how you maybe want to remember those. And cells can be connected by what we call desmosomes, gap junctions. Desmosomes form like a fibrous interlocking connection of cells. Okay, so we see a desmosome here, where we've got, this is a cell, and then this is another cell, and we can see they're connected with a desmosome. We have like what it calls a spot desmosome. So that's going to be a really tight connection. You're probably not going to get a heck of a lot of passage of material. You may get some with a belt desmosome, but these are generally going to be for stability or stabilizing the cells as they're connected together. So this would be a tissue, right? A bunch of cells connected together. So this is a tissue of some sort. Then we have gap junctions. Gap junctions definitely are going to allow for some sort of transport between cells. You can even see we've got channels. So some cells, certainly a lot of our cells, are going to need to transport and transfer water, transfer other nutrients, ions, transfer antibodies and items we might need to fight infection. So if this cell's infected, this cell may have what it needs to help fight that infection. So a gap junction helps with that. We can get quick movement of material. Yeah, and then we have what they call a tight junction. And the tight junction, that's going to be almost like a suture line. You can see it almost looks like we've again providing support and stability. Okay, so gap junctions form tunnels. Okay. Okay. Tight junctions, again, joined by collars of tightly fused material. Okay. And molecules are not going to be able to go through a tight junction at all. Okay. They may be able to penetrate these desmosomes, but this is kind of to seal everything off so nothing gets through. And it'll provide, again, some sort of structural support. Okay. We're almost done. It's a lot, right? We made it through cell organelles. Now, I just want to talk briefly about a couple more things. The rest of this is a little bit more functional stuff. So we do cover this in advanced, but I do want to mention it. You will see it in your text. You will have a few questions on these as well. But I just want to talk about a couple of transport processes. How do we get items moved through these connections, especially these gap junctions? Okay, so where do we get items transported into cells, out of cells? We can either do it passively or actively. So the passive transport does not require energy. So no ATP is needed with passive transport. We would see diffusion is passive. Generally, it simply means we're going from high concentration to low concentration. So particles generally want to move from where there are a lot of them to where there are a lot less of them. We see this going on right now all the time with people. I mean, especially with social distancing, you walk into a room and there's a lot of people. You're supposed to be social distancing and kind of dispersing, diffusing, going to where there's a less concentrated amount of people. So it's a passive process. Just particles are going to move to where there are less particles. Okay? So we see that diffusion. There's something called simple diffusion, which is what we see here. There's something else called facilitated diffusion, where we need some sort of a helper. So we'll take a look at that in a little while. But the idea is we're trying to establish equilibrium. So the whole point of moving items is to establish equilibrium. Okay, again, simple diffusion, moving particles moving through a permeable membrane from high concentration to low concentration. Osmosis is also a passive process, but instead of moving the particles, we can dilute the area by moving the fluid or the water. So osmosis is simply moving fluid to dilute an area that has more particulate, whereas simple diffusion is moving the particulate to where there's more water. Okay, moving particulate to where it's less full of particulate. The particles want to disperse and diffuse. Okay, the other way we could get the ratio balance or equilibrium met with concentration is dumping a whole bunch of building more rooms or building a bigger swimming pool. I've got 100 people crammed into a pool. Well, I could either remove 50 of them and disperse them out into other pools. That's diffusion. Or I can just build a bigger pool and add more water in and add more water and have a bigger pool, and the number of people doesn't change, but the amount of water does. Either way, they consider those to be passive processes. And again, we study this quite extensively in advance. So if this is feeling a little bit much after going through all this cell stuff, I totally understand. But I am going to keep trudging forward just so we can, you guys can get a move on and enjoy the rest of your day. Okay, so we've got ions. Okay, Generally, they're not going to be able to simply diffuse. They need some sort of a channel. So we get these small uncharged particles, oxygen, carbon dioxide, the gases. They're the ones that are able to go through simple diffusion. So we see this in thin layers. Larger uncharged molecules or particles, some water is going to be able to go through regarding osmosis, right? We can use water for osmosis, but like glucose and other sugars, and then these ions or charged particles, they're not able to diffuse. Okay. Osmotic pressure, we're not going to get into at all. This is an advanced concept. I do want to mention that the effect of osmosis on cells. So when we have this, what we call iso, meaning same, the tonicity has to do with the amount of stuff in the solution. So how much salt, for instance, sodium is in the water outside of the cell. If there's a lot of sodium outside of the cell, we call that a hypertonic solution. So this hyper means a lot of, right? Tonicity has to do with the solution. Okay, so a lot of solute in the solution. So what does the cell do? The cell goes through osmosis, and the cell's actually going to release water out from inside the cell. Remember, your cells have a ton of water inside of them. So we're going to see water released from inside the cell to outside of the cell to dilute all of that excess solute or sodium. So this is a hypertonic solution. We've got water then leaving the cell to dilute the area. So hypertonicity causes cells to shrink. If I have a hypotonic, so now we've got more sodium inside the cell than outside, so the water is going to leave the outside of the cell and go inside of the cell to try and dilute the excess amount of sodium, for instance, if we're using sodium. So that's going to cause the cell to explode or lyse. Okay. So water always travels towards where there's more substance because it wants to dilute the area. When we reach equilibrium, we have an isotonic situation. Okay, just a couple other things. Facilitated diffusion simply means we need a helper for diffusion to take place. We generally see this with larger molecules like glucose is going to need an integral membrane protein to help facilitate its ability to diffuse. Insulin is one of the primary ingredients to enable glucose to diffuse across a membrane. Okay. And again, we're not going to, there's other types of facilitated diffusion that deal with voltage, electricity. Again, that gets into too much with advanced, so I don't want to get into that. Lastly are the aquaporins. That just means a pore for water to pass through. Okay. So here are those channels. And again, membrane carriers. This would be like a helper. This could be glucose. Glucose comes in, connects with this membrane carrier, and then it allows it. It facilitates the diffusion. Okay. And again, these are passive processes. Diffusion, both simple and facilitated, and osmosis. These are passive, no ATP required. The active processes, active transport process, you can even see ATP in there. Now, that's not what ATP stands for. Okay, adenosine triphosphate, ATP, but we also could relate active transport processes to ATP. It's just a helpful kind of hint for remembering that ATP and active transport go together. And that typically means we need a pump or a vehicle, right? A vesicle or a vehicle needs gasoline. So when we go back to the Golgi and we look at the lysosomes and we look at some of these transport vesicles, they may need some sort of ATP, right? They need gas to drive around. And then certainly the pumps. We see calcium pumps, sodium and potassium pumps. Pumps come into play and active transport comes into play when we're going against our concentration gradient. So back to the idea of I've got a ton of people in the pool. Instead of building a new pool or removing some of the people, we force more people into the pool potentially. So that's when the pump comes in, ATP. Okay, so here's calcium pumping back against it. If calcium gets released to get a muscle to contract, we have to, at some point the muscle has to relax. So we need to pump the calcium back to where it kind of rests or lives between muscle contractions. And we need ATP to do that because the calcium is going back to where it's stored. It's going back home where there's going to still be a lot of calcium stored in there. So we are pumping calcium during a muscle relaxation against its concentration gradient, simply meaning we're pumping calcium from low concentration to high concentration. That requires extra effort. We see the same thing with sodium and potassium. We have to, sodium generally lives outside of a cell, potassium lives inside of a cell. So when I need to get sodium out of the cell and back to where most of it is, I have to use a pump because there's always more sodium out here. And so these three aren't naturally going to diffuse because they can't, because their molecules don't go against their concentration gradient unless we involve a pump. So we can pump three sodiums out, and then we bring and pump 2 potassiums back in. So we've got, and because potassium lives inside of the cell, so potassium needs to be pumped against its concentration gradient. Again, this is all advanced IMP stuff with a little bit of intro to the physical features. Okay. The physical feature for active transport of cellular transport mechanisms is a sodium potassium pump, for example. Pumping 3 sodiums out, pumping 2 potassiums back in. Again, active transport processes. We also have transport by vesicles or vehicles, which I mentioned a few minutes ago. That gets into endocytosis, or eating and bringing things into the cell. We have a particle, we can bring things in. We've got this little vehicle that came out and picked up the particle. This requires ATP. If this particle is something we don't need or want, we can destroy it with the help of some lysosomes or peroxisomes. And we also have the transport of whatever enzymes or membrane bound items that we need to transport out of the cell. This is what we call or exit cytosis. Okay? Exocytosis. Both of these transport mechanisms, because they involve a vehicle, require gasoline or ATP. Eating and drinking, by the way, endo, bringing in, if we're bringing in large particles, it's phagocytosis. If we're bringing in fluids, we call it pinocytosis, just as a side note. Whew, that's a lot to absorb today. Let's see, we're at about 115, 120 area. Cell growth and development. Let's take a quick peek at that. Again, this information, the rest of this is explored in more detail in advanced. So if there are things that you're kind of like, I don't know, it's okay. You'll have a few items from here that you need to know. Cell growth is a cell's lifespan is called interphase. Okay, the stage of the cell from when it's basically been divided and we have a new cell, that cell is now in what we call interphase. Interphase is from birth back around to when that cell is going to divide or replicate into two new cells. So interphase is in essence the lifespan of the cell. It has two distinct phases within it at the very beginning. We call it the G1 phase. So when we look at a cell, so a cell has just been born here, G1 simply means first growth phase. Okay. Now, as that cell's going through interphase, it may need some maintenance. It may need to be repaired. It may not be ready for its code to be replicated. We may not want photocopies yet of this cell because it's lacking maturity or it's lacking something. So we pull it out and it goes through what we call the GO phase, the maintenance phase. And G0 is what it really means. No go, nothing's going on. That's what the G0 or GO phase is of the cell's lifespan. Okay, but if it makes it, it gets maintenance is great, or maybe I don't need it. It could get pulled off the line because we've got plenty of cells just like it. We don't want to overdo things. Okay. Something called apoptosis is like basically the cell's death schedule. So apoptosis is programmed cell death. Okay, apoptosis, apoptosis. Okay, that's programmed cell death. So this cell could be programmed to go through apoptosis before it ever replicates. Okay. So anyway, G1 phase, first growth phase, GO phase, maintenance, maybe getting pulled off the line. Not all cells are going to go through the GO or the G0 phase. Some of them go right from G1 phase into what we call the S phase. And this is where we get the photocopying and the replication of the DNA. So DNA replication occurs during the S phase. By now, if we've made it into the S phase, we're likely going to get an exact we're going to get exact replicas of this parent cell here. Okay, I say likely because there's almost a point of no return here. However, it doesn't show it up here, but at the G2 phase, we could potentially pull it off the line and stop the cell from reproducing. Because we've replicated the DNA down here, if there's a mutation in that DNA that we just photocopied, so now I'm making a bad copy, or I'm making a copy of a bad code. So now the code could get transferred then to, or it will get transferred to the daughter cells. So that's kind of what this maintenance or G0 phase is, to make sure that we're not allowing mutations to pass through and get replicated during S phase. All right, so in a healthy cell, we're going to go then into G2. It's kind of like the twilight years of the cell. And then it's going to enter into cell division. And these are the four primary phases of cell division we call mitosis. Okay, this is called mitosis, what we call prophase, PMAT. Okay, so interphase is first, right? That's the lifespan. The majority of a cell's lifespan is spent in interphase. If we were to look under a microscope at a bunch of cells, we would see most of them just look like mature cells with a nucleus. That's because they're all in interphase. It's only a tiny portion of the lifespan of the cell that it actually starts to divide, and we can see on a microscope or under a microscope what these different phases look like. So prophase is opening the nuclear envelope and kind of allowing the centrioles and centrosomes to get lined up at the poles and get the DNA and out of the envelope, if you will, or out of the nucleus. And then metaphase, we have these spindle fibers that are all lining up, and we can get these chromosomes lining up down the middle of the cell. And then during anaphase is when we're going to split the chromosomes and form ultimately in telophase 2 separate cells that are going to start walking away or moving away from one another, which is what we call cytokinesis. Okay, so that whole cell division phase is called M phase, the mitosis or mitotic phase, which again involves one cell growing old and then basically allowing its nucleus to open up, release its chromosomes that then line up down the middle of the cell and then divide into two separate groups of identical chromosomes because we replicated them down here in S phase. So half go one way, half go the other. We have a complete duplicate or twin cells from the parent. So we're going to have two daughter cells that pull apart in this cytokinese, means remember movement. So cells moving apart from one another. So telophase, you'll be able to see pretty much distinct division of the cytoplasm or getting closer. We call them cleavage lines or furrows, cleavage furrows in the cells where you see kind of that depression or ridge forming that's distinguishing the two cells from one another. So anyway, that's the lifespan of a cell in a nutshell. All right. So cell reproduction again is M phase. So the lifespan is the interphase. Once we get into the reproducing of the cell, we call it M phase. Okay. And again, prophase is first. That's where we open up the envelope to get the chromatids and chromosomes all separated. We're going to get those centrosomes and centrioles lined up at opposite ends of the poles. We can start seeing spindle fibers being constructed, and then those chromosomes line up down the middle in what we call metaphase. Meta being middle, the equator, and each of those chromatids are going to attach to a spindle fiber. Okay. They're called sister chromatids, and they're going to be separated during anaphase. It's two separate but identical pools of genetic information being pulled towards. So you have the centrosomes at each end of the poles, and then we just pull that genetic information, half of it toward one pole, the other half toward the other, and each half is identical information. So now I have two separate cells, and those we're going to start to see then separate cytoplasm, separate nuclear wall, nuclear membrane, separate cell membranes. Okay. Telophase, again, DNA then starts to return to its original form. And we see then, again, two what are almost looking like separate nucleus sections here during telophase. And then you can see the cleavage furrows forming right here. So we distinguish, we can distinctly see two separate cells. Whereas over here, so prophase, notice how dark that nucleus is. There's a lot of action. Nuclei always stay dark. So prophase, we broke open the nucleus. The nuclear envelope broke. So you can see there's usually a lot more action. There's a lot of color going on. And then in metaphase, what you're looking for is this dark lines right down the middle attached to these little spindles. Okay, so prophase. See a lot more color, a lot more action, a lot of squigglies going on, and then everything kind of lines up. And then anaphase is where we split, like a banana split. Okay, anaphase, right down the middle of those chromatids. And then we end up with what is going to be two distinct, because remember, these are the bundles of genetic info. So then these are going to be the new nucleus for each of these cells. And then ultimately we get cytokinesis occurring, and now we have two separate cells. And both those go through what we call interphase. And again, we may not need two, right? Otherwise these things, cells expand, exponentially. So we do need some programmed cell death and apoptosis to kind of limit the number of cells that just keep it growing and growing. So that's where we have that kind of maintenance phase where some of these cells will get pulled off the line. Kind of as a side note, we will see this at the end of this semester in the reproductive system. We also take a peek at it at the end of advanced, but meiosis or meiosis, some say meiosis, some say meiosis, but either way, it's not mitosis. There's no T in there. This has to do with a kind of an extra layer of mitosis. The gametes, which are the sperm cells and the egg cells, all of our human cells have 46 chromosomes. So we want to divide the sperm cell to having 23 chromosomes, and we want to make sure the egg cell has 23. So when the sperm and the egg meet together and the egg is fertilized and we have a zygote, a new being, we have 23 from the parent, the dad, 23 from the mom parent. We put those together, we get 46 total. We can't have a sperm cell with 46 meeting an egg cell with 46. So we have to have half and half, 23, 23. Meiosis is the process of making diploids or diploids, which are the 46ers, into haploids, which kind of sounds like half. So haploid cells. Okay, that's all that is. It's an extra process. We won't get into too much detail with it. This is as much detail as you're getting right now regarding that. Next, at the end of the semester, I'll touch on it again, but at the end of advanced is when we really get into that aspect of cell division, meiosis. Okay, there's only a couple slides left. Chemical reactions, we looked at this last week. Remember, we had synthesis and decomposition. Synthesis is building something up. We see this also called anabolism, like an anabolic reaction. So synthesis equals anabolic. We see protein synthesis, lipid synthesis. These are things that mean we're taking the little amino acids and we're stringing them together into big long chains or beads. Okay. Decomposition means we're taking the beads and we're breaking them apart. So when you eat something, you'll break those things apart, right? Your body is catalyzing or catabolizing or decomposing the big protein into smaller amino acids. Okay, those are decomposition reactions. And then and enzymes are what help to break those macro bits into smaller bits. So the big steak becomes a bunch of little amino acids. The big long necklace becomes a bunch of individual beads. Hey, that's what the digestive system's doing. And then the bloodstream takes the little beads and delivers those to the cells. And then the nucleus of the cell has whatever directives and recipe that it needs for the ribosomes and the rough ER to start stringing those beads back together in what we call a synthesis reaction or a protein synthesis. So that's that cycle of you are what you eat. Catabolism, again, breaking things down. Hydrolysis, we add water, it's going to break it down, right? We put the big chunk of steak into the guts, we add water, we add enzymes, and boom, we're going to get the breakdown into smaller bits. Usually the end products of catabolism cellularly are going to be CO2, water, and other waste products. Cellular respiration is a form of catabolism. We're breaking down glucose, ATP. We're getting glucose and oxygen, combining those initially, and then we get ATP. That catabolizing is going to lead to carbon dioxide being one of the waste products of cellular respiration. Okay, then so again, this is all advanced IMP stuff. But the state does want us to just basically tell you that metabolism is a chemical reaction. We talked about these last week. Catabolism breaks things down. Anabolism builds things up. Typically, dehydration is going on with anabolic reactions. We're actually removing water or going through what we call dehydration. Okay? That allows the small molecules to become bigger molecules. Okay? So anyway, FYI, really, I'll have a couple of questions, the very basic questions about metabolism, anabolism, and catabolism, or synthesis and dehydration. Okay, catabolism, with again, mitochondria, they want us to mention too that, which we already did earlier, cellular respiration, ATP. Okay, glucose and oxygen give us up to 36 ATP. And again, most of that's happening in the mitochondria. That's what we call aerobic respiration. And again, anabolism, building things up, putting all of the beads together to make a necklace. That's protein synthesis. So the central anabolic pathways in cells. So this does not involve anything more, anything less than ribosomes and RNA working together to put amino acids into either larger enzymes or larger bits of protein. And of course, we have lipid synthesis going on as well. And again, you want to remember to, we transcribe or make a photocopy of the DNA as RNA in the nucleus, and we translate that code in the cytoplasm via the ribosomes, rough ER, and those messenger and transfer RNAs. Okay, and then again, aerobic versus anaerobic. Aerobic is simply using oxygen to get more ATP. That takes place in the mitochondria. Anaerobic, oxygen deficit, get less ATP. Anaerobic, oftentimes then is going to take place in the cytoplasm of the cell. And again, more about this in advanced A&P. All right, you guys, we made it. It was a long, winding road to get here. But again, these last couple of bits, there'll just be a couple of very basic questions from these slides, and the bulk of your test bank is going to be being able understanding what structural aspects of the cells we have, what are some of the basic functions of cell structures. Other than that, that's pretty much it. Your portfolio assignment is similar to last week's. There's some definitions, a couple of items to break down, and a small case study. But I'll have that out for you later this afternoon. And then on Thursday, we're going to go through some visible body activities. So again, make sure you have that ready. What else? I'll have a little outline for you to access. Basically, Unit 3 or Week 3 is going to be open. For the folder will be available for you to access. And again, it's going to look a lot like what you saw last week. Padlet link, an outline, and then link to the slides, and then your visible body drop box that you'll need to, and again, I'll have directions for how you're going to go about that, and then your portfolio assignment and exam. Okay, so pretty straightforward with what we're doing and where we're going. And again, on Thursday, I'll speak to the visible body, and we're going to work on that together. In essence, there are a few things obviously you'll do on your own, and there'll be a small Dropbox. I believe, I'm not sure yet. That's what I'm still trying to kind of work through, because most of you do have the visible body. So some of the the points that, remember, the visible body grade book is separate, is different. So your visible body activity grade in your Blackboard grade book is 20 points every week. So you'll be doing either a drop box with a small kind of reflection on the visible body. It's probably what I'll do. But you'll still be doing some of that work in the visible body program. So I'll show you more. I'll explain it in more detail. You'll know what to do Thursday. It's not going to be anything intensive. It's just simply to add to what we did today. You have several of the foundational aspects of what cells are. You can go back and watch some of this. You can look at chapter 3 a little bit. Just kind of let it all sink in, I guess is what I'm saying. And then Thursday, I'm kind of going to go back through what we did today, just through the beginning stuff, especially, but using visible body. So you'll be able to see a little bit more structural stuff and three-dimensional stuff. We're just going to reaffirm and etch in some of that terminology we went through today. We're going to do on Thursday as well. So if some of this is a little bit mind-blowing, that's okay. It's supposed to be. That's why it's science. That's why it's school. It's why it's medical. And it's why it's a lot. So just know that you're going to get more this Thursday and throughout.